All right, so this week we're going to have a slightly different topic than last week, or that's been kind of over the last couple of weeks, which is that we're going to talk about particles and interactions that happen between aqueous solutes and particle surfaces. And we're going to highlight that process in estuaries. Process happens in other places, but it's like most pronounced in estuaries. So today we're going to talk a little bit about estuaries first, and then we'll talk about some of the phenomena that involve mixing of water of different types in estuaries, especially seawater and river water. And then we'll start talking about these sort of particle-solute interactions, and then we'll pick that up next time. And as you can imagine, particles come in all sorts of sizes and shapes and flavors, both natural and anthropogenic. So it's a broad topic, but what I hope to have you come away from this week with is kind of an idea about the complexities that we have in natural environments where, because of various kinds of interactions, acid-base, Lewis-base, charge transfer, ion exchange, all these kinds of things, which we'll go through in detail, that things are constantly evolving in any environment that has particles. And this will become important later. Also, we'll be talking about things like water treatment and sewage, because again, those have particles in them. So first, let's just talk a little bit about estuaries. It's not a topic that's covered in much detail in a textbook. So I just thought I would just remind you about what estuaries are and how they come in a wide range of shapes and sizes. So estuaries are the zone between which we have the ocean and a river, and estuaries can exist in places where the river flow is very variable throughout the year. Some places, even when rivers dry up in the dry season in a particular place, there can still be an estuary because there can be subterranean discharge of fresh water. And these are just two examples of estuaries that have a very different shape, especially this one. You can see how much sediment has been deposited, and highlighting the fact that we've got agricultural land going right up to it. So it turns out that because of the particles that come off and because of the sort of various other chemical processes that happen here, estuaries are places of very high biodiversity and high density, population density, in part because of kind of an extra rich food web supported by nutrient and particle flow coming off the continents. They also happen to be places where humans have chosen to put a lot of settlements, sometimes harbors and other things. So it's kind of an interesting interface zone that we'll learn more about. So it's another ternary diagram where, the closer you are to one of these corners, the more you're expressing that thing. These are the kind of three parameters that define how waters mix in estuaries. One of them is driven by the tides. So a place where there's a lot of tides or not so much tides is going to change how the dynamics of mixing and how that varies throughout the day or throughout the year. We have wave regime driven estuaries, and then ones that are driven by the river flow. And you can find estuaries around the world that have all those characteristics. Turns out here in Hawaii, we tend to be down in this corner. We don't have a big tidal variation, and we don't have very big river flow rates. That doesn't mean that during a storm event or something like that, where a river is flowing very strongly, that an estuary might migrate somewhere in this diagram. But this is basically where we are. And you can see some, some well-known, some rivers. I don't know where all these rivers are. It just gives you an idea of what the main forcing functions are. So here's a few more satellite images showing you different kinds of estuaries. You can see these very braided rivers coming in. That's obviously a false color image, satellite image, but again, you can see the farmland going right up to the river mouth. You can see the sedimentation, the sediment flow, and in fact, one of the things that we find in estuaries is that we can find more sediment here than we find in the river. And part of this is due to the processes we will talk about today, which is the release of particles from solution, these hyperfine colloids that I've talked about before. There are things about the introduction of salt water which destabilize the colloids, cause them to clump together and fall to the sediment interface, bringing with them a bunch of dissolved solutes. And so that's kind of an important aspect of everything that happens there. So this is just, Manahan's kind of one statement about estuaries, about how they're kind of important and breeding grounds of a lot of life, and then they're not discussed anymore, which is, as I say, kind of unfortunate because for an environmental scientist, estuaries are hotspots of a whole bunch of different chemical interactions and interactions with food webs. And this is, an example of a compound estuary that we have here on this island in Oahu. This is, you're looking towards the Ko'olau across the kind of northern part of Kaneohe Bay. And so there are rivers that come down through several of these divides. They're relatively small rivers, but you can also see these have been modified, right? Modified by, in this case, fish ponds, superimposed upon the pre-existing coral reef structure. All of these things affect the way waters mix and the dynamics of mixing and the distribution of solutes and particulates. And so that's not uncommon. A lot of estuaries are modified around the world. So I just point out, I have a few slides in here if you're interested. There's a EPA report that you can look up that sort of discusses a little bit of stuff about estuaries in Hawaii. You can get it on the web. I've linked it here. And I'm just going to click through a couple of slides really quickly. So they studied all of these different estuaries. And as you look at them, you can start to think about what would make them different, being on the windward side versus the leeward side, being exposed to big waves in the winter or not, being part of a big harbor system or draining particularly right out into the ocean, being in a place that's been completely modified, like this area around the airport or this area out in Hawaii Kai, so that, whatever the characteristics of mixing and flow are, they're different, perhaps, these out here on Barbara's point as well, than they would have been previously. And they do a lot of detail. These are just sampling sites that you can look up. I'm going to show you some plots just so you can see what some of these parameters look like. They also discuss some estuaries, one of them on the south shore of Molokai and a bunch on Manoa. So, and if you want to dig up. So the thing about estuaries is that we have this mixing of saltwater and freshwater. And there's a lot of different aspects to that, but I've kind of summarized some of the kind of important chemical things, right? So we know seawater is basic, pretty much everywhere, and river water is usually neutral to acidic. There are a few places where this deviates, but this is kind of a general thing. The total dissolved solids in seawater is pretty constant, plus or minus about 1 per mill unit here at about 35, and river water is a lot less. 5 to maybe 10 parts per thousand. But the important part is that whereas the ratio of all the salts to each other in seawater are constant because they're based on conservative elements, river water comes with different things in it. So that for seawater, we have a constant chlorinity, which is just the amount of chlorine in parts per thousand as chloride ion. And you can see it's the biggest part of the total dissolved solids in seawater. And in river water, especially pristine, uncontaminated river water, this is 0. You go to places where they salt the road and stuff, you can find a little bit of chlorine in there. But what we tend to track the mixing of these two components by looking at things that have 0 to 19. It's more reliable than picking salinity. But you will see a lot of papers and whatever authors using an assumption of salinity of 0, which is not such a great assumption for most rivers. The other thing is that the sediment load is very high in rivers and kind of low in general in seawater. When we get into the coastal zone and we have waves and stuff, that can be higher than we expect in the open ocean, but it's still relatively low. So then we have these physical things, which we've already kind of talked about a little bit, but we especially want to highlight things like the flow rate of the river, the suspended particle load in the river, the shape of the estuary, which is going to determine where and how things mix, and the local tidal fluctuations. Okay, so these are some diagrams. This is a diagram plotted of salinity in parts per thousand versus the sort of cumulative percent area. It's kind of a funny diagram, but it's basically saying that if we go into a particular estuary, and this is the summary of all the different Hawaiian estuaries kind of superimposed here, that the vast majority of, by area space looking down at plan view of our estuaries are almost like pure seawater, right? like this thing starts to tick up at about 20%. So only about 20% of the area of our estuaries is anything resembling kind of river water. And this is largely this function of being kind of driven by the tides as opposed to river flow. If we went to a place where there was a huge river flow variation and especially a robust flow, we would see a bigger area being dominated by these kind of low salinity zones. This is just a variation in temperature. You know, the solubility of chemicals are temperature dependent. We tend to have relatively warm runoff compared to our seawater, which is also relatively warm. So we have a pretty small temperature variation. And you can see that that mixes more kind of quasi-linearly between sort of, you know, average river water and average seawater for the area, which means that the parameters that control how temperature is distributed and how salt content is distributed are somewhat decoupled, which is not an uncommon thing in oceanographic systems. This is pH, and again, we go from sort of, this is seawater pH, and this is the average pH of our river waters. Our rivers tend to be much less acidic. This is in part because the water flow doesn't have very much time to acquire those attributes that we get from exchange with the atmosphere that help increase the acidity. So on average, our acidity isn't that low compared to other places on land, but nevertheless, you can still see this kind of mixing, and where most of it happens, the sort of maybe 40% or so is more river-like, and the rest is more ocean-like. And then this is sort of the depth, which is another interesting characteristic. So our estuaries tend to be kind of shallow. These are depths in meters, and so you can kind of see the distribution of that where, again, you know, sort of, they're all throughout the range there, but relatively shallow. Okay, so this is a plan view map showing you 4 sampling stations going from a river to the sea up on the top. That means someone went out with a little boat and a series of water bottles, and they dropped them down on a wire, and they took water from different depths. Then we plot them up on these kind of diagrams, which are salt content versus depth, okay? So this is what you would find in the water with depth. And this is an interpretation of the flow parameters that would cause that. So that, and we talked a little bit about this last time, this is the example of the Thames River, where the mixing is basically very much vertically distributed, so that we go through a salt gradient, up a salt gradient from river to ocean, and it doesn't vary with depth. Now, the kind of more extreme condition would be something like this, where we have really strong tides that are pushing in water across the bottom, coupled oftentimes with a very strong river flow, which is pushing water across the top. So we get more of a horizontal stratification. So that when we go to sample our waters, as we're going down, we might find different saltiness in the surface water, but the bottom water is always really salty. So you can imagine, I mean, this has salt water all the way back to the very deep into the estuary at its deep waters, and this has fresh water. And these are somewhere in between, and this kind of illustrates this kind of pattern where when we have a gentle competition between flow, we oftentimes have an upwelling current that causes some of the mixing to take place. And this is a salt wedge estuary, which again, I showed you a diagram for last time. And each one of these things, what's important, has a different location where the saltwater and the freshwater mix. So all the stuff we talked about, particles today and tomorrow, are going to vary physically where it happens in the estuary, meaning is it going to happen all the way to station 1, it's going to happen in station 4, it's going to happen throughout for our particular estuary, and therefore where might we expect to see the legacy of those things in both the water and the sediments at the bottom. Okay, so this brings up this notion of circulation patterns. These are plan view, cross-sectional views cut through a river going out to the sea. This is the typical pattern of estuarine circulation, meaning deep water comes in around the bottom of the estuary, competing with river water, and there's an upwelling mixing current. And this can tend to bring with it some of the characteristics that seawater has with it, and introduce it into the shallow photic zone here. There's oftentimes a lot of sediment associated with this, as I've talked about. So we don't tend to find much sort of enhancements in photosynthesis associated with this process. We tend to actually find it being suppressed because of the particulate load, but you can find estuaries that contradict that. Another interesting thing is that one category of estuaries that has something that we call a sill, which is an obstruction at depth that impedes flow, can't mix this way because the ocean water can't come in along the bottom. Classic example of this would be a terminal moraine in a fjord, right, which was carved out by a glacier, and then the glacier retreats and leaves a pile of stuff there. That's one way to cause it. You can have it caused by various other things as well. But in these cases, the mixing is usually really different. Because we can't have water flow like this, we have some sort of competition of flow. Whatever's being tidally and wave driven on this side, then whatever is being driven by the river. And these tend to mix in the opposite kind of direction. So this pattern for those two types of estuaries give us these kind of different level of stratification where water spends some time and has different characteristics as we go either with depth or laterally. And so some estuaries act like they're stratified kind of like a lake, and some of them act kind of like they're stratified like a lake on its side. Just go back to these examples and think about that, right? This one here is stratified like a lake. It's less dense and more dense. And this one here is like stratified like a lake on its side. But because we have stratification, we can induce the same sorts of things that we find in other stratified bodies of water, which is differences in chemistry. In a case like this, these bottom waters become isolated from the atmosphere, just like they would be in a lake. And so if water spends enough time there, it has the possibility of doing things like consuming all the oxygen and going down the redox ladder. Something like this is like a lake on its side. It still has exchange with the atmosphere in all of its zones. So it doesn't tend to get those same kinds of effects. Okay, so here's some more kind of estuary categories to think about. And you can kind of imagine some of these and how they might apply, like here, for instance, in Hawaii. There's a combination of sort of what is the shape of the estuary, what's the flow of the river, what is the kind of wave regime that you're coming into, how much sediment deposited there is. So for instance, something like this, a blind estuary, great example of this would be Waimea Bay, right, where we see the water coming in and all of a sudden we get that big sandy beach. The exchange between salt water and fresh water, it's not that it's being stopped by that sediment, it's just happening below where we can see it, right? And it's happening in a much impeded way, but it still happens. So each one of these kinds of estuaries, we might predict to find different places, and especially geographically, but also during the year, seasonally, where the mixing happens and where the chemical anomalies happen. So now let's just think a little bit about sediments and particles themselves. If you think about it, the sediment load that comes off the continents, which we've talked about already, or islands such as ours, are basically a significant fraction of the chemical burden that rivers bring to the ocean, especially when they're draining through anthropogenically induced or perturbed, I should say, landscapes. But even in a natural sense, a lot of these particles come down, right? It's something like 80% of their chemical load is, for most elements and most chemicals, is as particles, and that tends to mostly be deposited in estuaries, right? So estuaries are a place where a lot of the stuff builds up. And this ends up being the place where a lot of the particulate load that comes from human activities also tends to accumulate. And if you put the intersection of that with the kind of intersection that we talked about before about the sort of biological diversity and high population density that we tend to find there, along with the fact that a lot of human population centers have developed around estuaries, you have the potential at least to introduce a lot of extra contaminant into food webs that then interact with humans, and they are a place of great concern for that reason. Okay, so this is just some kind of examples of what sediment transport looks like. So this is, estuary in Africa. There's two of them right next to each other, right? So you can see this very complicated low flow river system with a very sort of crenulated coastline and this high flow river right next to it. And you can see the vast difference in the amount of sediment coming down. the amount of particulate burden. This is just a false color image, kind of highlighting this yellowy color is less clear and this bluish color is more clear. And so as you might imagine, even if everything else about these things, the tide regime and the wave regime, are the same, you're going to find differences in what is able to come out of this river and get into the nearshore region in both of these cases as a function of these particle interactions that we talked about. And this is kind of an extreme example. So when we look in an estuary, and we can look today and we can make samples, we can also go into the past, look into the sediments and get a record of how things may have varied. A good example is, for instance, pre-human settlements and post-human settlements in various places where this is easy to check, because we can find a legacy of this mixing down in the sediment record, right? And so this is, again, walking up a river from out in the ocean to over to up in the river. And then down here is plotted the different kinds of sediment deposits that we expect to see. And these sediment deposits are complicated, especially in estuaries that have human activities. Think about a harbor where we're dredging or whatever. But nevertheless, there are some patterns that we can deduce, such as if you go all the way upriver, you have what we call fluvial sediments, sediments that kind of reflect the river itself. And as you can imagine, when you come down from kind of a narrow constricted body of river water into an estuary, the flow starts to slow down and broaden out. And so what we tend to do is deposit our more coarse fluvial or riverine particles far upriver. And as we go down, the water is able to carry less and less of these particles from a size perspective. So we get fining. Okay, we get fluvial particle fining. And these deposits, especially when we're up in the river mouth where it's still relatively high flow, we can get what we call cross-bedding patterns in the sediments where we have little packets that are overlapping with each other. And as we tend to come out and we get to these sort of finer particles, we tend to see more laminated sediments, sort of flat line sediments. So when we come to the ocean side, we see the same thing. We'll see primarily sands and sand-sized particles, sometimes coarser particles that reflect whatever we find on the coastline. And then, moving inland, again, we can see the same kind of cross-bedding of the finer bits of particle that can be carried in by either the tides or the wave regime. And then somewhere in between, we have the zone of mixing. Okay, and the zone of mixing of waters, and we find mixed sands there. And the sediments that we find there can also have some other really interesting characteristics. They tend to be the finest particles, right? The stuff that couldn't get carried in very far this way or this way. They tend to have a lot of organic matter, which means that they tend to go reducing very close to their water interface, because a lot of the organic particles tend to be on the fine grain size. And there tend to be other kinds of disruptions that we can find in the sediment, such as bioturbation, which is organisms sort of burrowing in and disturbing the sediment because of the high concentration of organic matter and high biomass. So in addition to there being kind of patterns that happen in the water, there are patterns that also happen in the sediments. And when we go to try and preserve chemical records by drilling down into the sediments, we tend to get a more kind of straightforward and reliable interpretation when we're over here or over here. When we get into the center, we can see changes that might reflect, how the zone of mixing may have migrated with time, but we also get superimposed with other processes like redox ladder processes and bioturbation processes, which make estuarine sediments complicated, but nevertheless important to study. So our estuaries tend to be dominated by pretty fine particles compared to others. This is just showing you the sort of percentage of particles in the silt and clay fraction. You can see here in the sort of urbanized and unurbanized, these are urbanized and these are unurbanized. In the urbanized areas where we have hardened pathways, the water flow rate is increased and the sediment load tends to be decreased, we've sort of changed that natural pattern of having lots of very fine particles to having not as many fine particles. And so that makes some difference in the sort of chemical load that these things have. This is just a few more measurements. Our estuaries tend to be relatively high in turbidity. Again, it's because of the small particles. The urbanized estuaries are a little bit less turbid. And those are important parameters. Those fine particles are the ones that contribute the most to the kind of chemical mixing stuff we're going to talk about next. Okay, so now let's think about a little bit what happens chemically when we mix water and then how these particles superimpose on that. So if you think about river waters have different things dissolved in the seawater, and we'll just think about the major components of what's coming off the landscape. Rivers generally have more iron and aluminum, nutrient elements, silicon, and dissolved organic carbon. And seawater has more of these kind of major salt ions. And when they mix, if there was no chemical process affecting any of them from the mixing, they have a process that we call conservative mixing. That's just where you mix, for instance, equal proportions of one thing and another, then the mixture is going to have a 50-50 mix of all those concentrations. But in other cases, the very fact that we're mixing things causes things to happen that changes the solubility of stuff, right? And so that will be a chemical process that happens within the estuary. And we're going to define that on the next couple of slides in a second. But these processes can take one of two forms. They can make things less soluble, in which case they go down to the sediments, or in some cases they can make things more soluble, in which case they come up into the water in increasing concentration relative to what we would expect by this conservative mixing. And we also have processes that happen in the nearshore region, and we'll highlight some of those next time. That all the action doesn't happen just in the estuary. Some of the chemical processes happen in the coastal zone. But these kind of three things combine to make average river water and average ocean water so different, pretty much everywhere on the globe. Okay, so the next thing is talk about a little bit our particles, right? We've talked a little bit about colloids before, but I want you to think about them now physically as materials. These are things that act like they're dissolved. They're solvated. They're not actually dissolved. They're just so small you can't see them with the naked eye. And they've got these charges on their surfaces, and these charges help them stay in solution under certain conditions. We call that solvated. There are other conditions that destabilize the processes that keep them in solution. They tend to stick together, clump together, and then fall out, and we call that desolvated. And there's various names for that process, which we'll get into in a second. But these are artificial colloids that people make so they can make them very reproducible in different sizes and shapes, and then study them. And so these are different colloid particles, and you can see the amount of magnification here: 50,000 times, 100,000 times. These are tiny. So that's a 100 micron scale bar. So colloids generally are in the less than 2 micron category, oftentimes smaller than a half a micron. A micron being a millionth of a meter or a thousandth of a millimeter. And I would say kind of going up maybe until the mid-70s or mid-80s, when people were studying river water, they would filter it with a 2 micron filter and then look at the composition of the water and the composition of the particles without recognizing that there's a lot of particles that are smaller than 2 microns that are holding things on their surfaces that give you a false sense of what's actually in the water. We now understand that and have protocols that allow us to look at smaller and smaller particles. But in any event, they can be a very significant control on solution composition. Okay, so now let's think a little bit about those particles. So those particles have chemical entities on them that can accept charge, and whether it's an ionic charge or Lewis acid Lewis base kind of interaction, and that causes chemical reactions between the particle and the solution that affect both the chemistry of the water and the chemistry of the particle. And those are especially prevalent in that colloidal fraction, those really, really small particles, which exist in fresh waters especially, but don't exist as much in salt waters because the salt destabilizes them. And we'll talk about that process in a second. So this is just a chart showing you kind of size distribution within the particulate load up here, going from sort of atoms and molecules, viruses, You can see where bacteria and algae are. Colloids are the yellow field. And then we get into like the proper particles. So this is kind of on average. They're big ranges, but you can see the sort of size fractions here. And what determines whether something's going to be a colloid or a suspended particle is a combination of how big is the thing and what is the density on its surface of the chemical entities that allow it to take a charge and then stay solvated and make it into a colloid. So a really small particle without much of this stuff on it might, fall out of solution, whereas a bigger particle with more of these chemical entities on it might stay in solution. So it's given as a broad range. It's just across the bottom of the kinds of things one would use to filter out those various things. This is just a slightly bigger kind of another diagram with a little bit more information on it with different kinds of particles. It includes particles that are in water as well as particles that are in the atmosphere. For instance, smoke, tobacco smoke, for whatever. And the important thing to note is that there are a lot of different types of particles in the environment, right? We're only going to talk in the generic sense today, but they're the particles of nature which come from, for instance, the biosphere and the geosphere, and then there are the particles of the anthosphere, which are many and varied. All sorts of different things that we use all the time produce particles which end up getting into the ocean. So this is another picture of a colloid. This is an iron oxide colloid. Again, a commercially produced one used in things like water purification. But hopefully you can see they're very small. That's a 10 micron scale bar. So many of these, obviously, a micron or less, with these kind of fluffy edges, very broad, high surface area. But as we'll talk about in a second, there are things that can cause these to clump together and pull stuff that's stuck onto their surfaces out of solution. And that really happens a lot in estuaries. Okay, so the particles that make up colloids, and in this example, we're talking about clay particles. They're natural particles. Clay particles take usually a negative charge on their surfaces, and that means they attract positive ions to them, cations. And this process is part of the thing that allows them to stay in solution, solvated, whereas a similar size particle, similar size and density particle that doesn't have the same chemical property would just sink. It's the charge that allows it to stay in solution. But there are things that can happen that cause these particles to stick together and clump. It's a process we call flocculation, so that it makes kind of a bigger particle. And you can see that this kind of blue zone, which is the zone of attraction of cations to the surfaces, has gotten a lot physically smaller. One of the things that causes this to happen is introduction of salt water. So if you got fresh water, you can have, and it's mixing with salt water and estuary, you'll find this kind of zone of cation cloud around the particle surfaces collapsing, which causes the particles to sink together. And anything that's physically stuck to their surfaces, think about heavy metals, think about organic carbon chemicals, for instance, they're going to get sedimented out in this process of flocculation. And they're going to show up in that zone of mixing in the estuary between where the riverine and the marine sediments are most known. So that can be a very important phenomenon. So this is just a little bit more about how that process happens. So there are electronic forces, Coulombic forces that help solvate or allow these particles to kind of defy gravity and stay distributed within the water. They're not dissolved, but they're distributed. They act like it. And so what we're really looking at is a competition between forces that want to pull something out of solution by gravity and things that want to keep it in solution by this Coulombic interaction. And so that Coulombic interaction is completely dependent on charge density. We'll talk about the way in which these particles get their charge in a second. There's a couple of different mechanisms, and all of those mechanisms tend to work in river water and not work in salt water, so that in the mixing zone of the estuary, we basically lose our solvation, gravitation starts to take over, and we flocculate our particles. That brings a lot of chemistry down into the water. And again, that part of what makes colloids so important is the fact that these things only work when the surface area is high relative to the mass of the particle. When the particle starts to get really high, then the gravitational forces, or when the particle mass gets high, the gravitational forces win out. but when they're small, they can stay solvated for a much lower amount of density of charge. So this is just some terminology about colloids. And, you know, when we have colloid particles in solution that are solvated, those are officially called sols. Not that many people use that term, but one of the things that can happen with sols is they can self-organize into something called a micelle. And this is a picture from your text. And we've already talked about long chain fatty acids before. This is the anionic form of it, the carboxylate form. And that carboxylate is soluble in water, takes a negative charge. But this whole long chain fatty acid hydrocarbon component is not soluble. And so another way to make a colloid is for these molecules to kind of self-organize, where each one of these things is sort of depicted as a little chain here. They get together, clump up, and put their water-soluble end to the water and the fat-soluble end inside. And in fact, this is the main way in which triglycerides are transported through your blood, is in structures just like this. So this is particularly important when we're talking about organic carbon that can be colloidally suspended. And so if you can take a colloid, if you're really careful in the lab, or you know it's been perfected in the production of things like Jell-O, if you take water away from it, you can make what's called a gel. And that, if you dry it enough, you will make it a solid. But there's some water that is chemically bound in with the particulates that is associated with it that helps with this process of flocculation. And they're only stable at certain conditions, and it totally depends on the particular particle content, but especially the pH is really important, and the total dissolved solids are important. You can find examples where changing the PE a lot can change the solubility of, or not the solubility, the solvation of colloids, but it's not nearly as common. That's why I put it in parentheses. And when they become unstable, when the conditions change, the destabilized colloids are flocculated. That's the main name we use for this process in nature for the removal of particles from solution. Okay, and so this is just a little bit more about kind of that process. There, you will find different terminology applied in different kind of literature, and there are slight differences between the process of flocculation, agglomeration, and coagulation. Agglomeration and coagulation are terms that are more common when we talk about water treatment. But they're all in essence the same thing, which is, or I will use them synonymously, which is the taking of suspended colloidal particles, having them stick together because some kind of coagulant or solvation destabilizing agent, such as salt in the case of estuary, is introduced into the water, causes these particles to clump together and sink out. And anything that's stuck with those particles goes down to the center. So that's in essence what the process of flocculation is. So flocculation is enhanced by several things. One of them is changing the flow field of the water. Make water slow, especially over a really strong gradient. That causes flocculation. So it's not like it's especially a rapid change in flow field, such as a river flowing into an estuary. If you change the ionic strength of the solution, ionic strength is a way of measuring salt content. It's basically the kind of molality times the charge squared. So that like a cation that's got a +2 charge has a bigger effect than a singly charged ion. The sum of all those in solution divided by 2 to account for the cations and anions. I'll give you that equation later. It's not super important, but it's just ionic strength is a measure of saltiness. And so these are the two primary ways in nature. You can also do it by evaporating a bunch of water, which then in effect increases the TDS. Or you can introduce a chemical destabilizing agent. And in the case of rivers and estuaries, the salt is the destabilizing agent, but it's doing this. There are other destabilizing agents, especially that can be employed in water purification, organic or inorganic, that can help make that happen, not because of salt and ionic strength, but because of other things that affect surfaces. So now let's think a little bit about how charge is distributed on surfaces. So these are a couple of depictions of the two end members of a particle and what's happening in the water adjacent to the particle. So we've got this model here and this model. This is called a single layer model, and this is called a double layer model. Double layer models have a fixed layer on them. Single layer models don't. So what does that mean? In this example, we've got a negatively charged particle. Let's call it a clay. And that particle presents itself to solution and it naturally attracts cations to it. Now, you don't get a pure separation of cations from anions. Instead, what happens is that it attracts more cations to it than anions. And importantly, just because a particle might have a net charge that's negative, it doesn't mean every little thing on that particle is negatively charged. It just means in net. So you can have positive and negative chemical moieties on the particle, so that when we look in distance away from the particle in solution, next to a negatively charged surface, we see a higher number of cations and anions kind of decreasing as we go out into solution. So it's attracted some stuff to it, a little bit more of this because it's a negatively charged thing. And these kinds of cations and anions can exchange really easily with solution because they're not stuck to the particle surface. And this sort of relationship is part of what keeps the thing solvated, keeps it from being gravitationally settled. In a double layer model, we have a layer that is really stuck, right? So some cations are drawn to that surface and they are there physically. And then when we get beyond that layer, we still have this kind of similar pattern like this, although only the cations are shown and not the anions. These tend to be stuck more, specifically to this particle surface and are not as easy to exchange. And there's a whole bunch of more complicated models in between, and they depend on things like the size and shape of the particle and the chemistry of the particle. How does it have its charge? What is the thing that's causing it to be charged? And you can have negatively charged particles or positively charged particles, and they would do sort of the opposite thing. Also, sometimes you'll see people talk about the stern and gooey layers. The stern layer is the stuff that's stuck to the particle. This is like a little bit more detail about that. Again, sort of 4 different types of these fixed layer models, and what they have, sorry about that, the distribution between kind of particle density on the, charge density on the particle surface and what it has in the water. And it's not so important to know these details. I just wanted to point out to you that it's complicated and it depends a lot on the individual particles. But in a net sense, I think we just want to think about this as some particles carry with them some charges that are stuck onto them very strongly. And when we have those kinds of associations, they're going to cause really predictable things in water. Other particles can take sort of stuff stuck onto the surface that depend a lot on the local conditions. So sometimes it will have an effect and sometimes they won't. Okay, so those ionic clouds, the things that stick around the particle surfaces, can change their dimensions. They can expand or collapse as a function of solution properties, especially how acid or base things are and how salty they are. And so if we start to get too much ion cloud interaction, then we desolvate and flocculate. So there's a couple ways of doing that. One way to think about it is, let's say we have so many particles that they're bumping into each other, right? They can desolvate themselves. And or we have one of these sort of chemical destabilizing agents, like changing the pH or changing the salt content. But another thing to think about, and this will become important later when we talk about groundwater chemistry, we can also have so much particle that we have more particle than water in a system, right? And so in groundwater, we have solids, and those solids are fixed in place. They don't get to move around, but their surfaces take charges just like this. Pretty much every surface that you find out in nature has charges on it. And when we pack them together very closely, and their ionic clouds from one side of a pore to another interact with each other, then they can actually prevent ions from flowing through that system. And so they can cause a process that we call chemical retardation, which makes some chemical solutes in the water flow much more slowly than the water itself. And so we'll talk about that later in the semester, but it's from the same fundamental process of particles taking charges on their surface. Okay, so now let's talk about the 3 main sources. The primary mechanism are acid-base reactions. Acid-base reactions of the Bronsted type, things getting protonated, things getting deprotonated. We also have ion exchange reactions. That's where we substitute one chemical for another. And if the two chemicals we substitute don't carry the same charge with them, that can create a charge imbalance. And the third one are Lewis acid, Lewis base interactions. And these become particularly important when we want to sorb things like organic matter onto a particle surface. And so colloids themselves can be made from particulate organic matter or inorganic. And there are certain characteristic charges that they take. Like if we have non-dissolved particulate organic colloid that's very rich in humic substances that have a lot of carboxylate ions on them. It's not dissolved, but those carboxylate ions are negative, and that gives that particle a negative charge, so it's going to attract cations. Clay particles also tend to have negative charges. It's common to find negatively charged particles, but we also, as you'll see in a second, can find positively charged particles. Many of the particles that take a positive charge take it because of this process, but some of them take a positive charge because of this, because they can protonate. They can take a hydrogen ion from solution, act like a base, and then have a positive charge and attract negative charges too. Okay, so I'm going to go through each one of these three really briefly. So in the case of acid-base reactions, every little chemical entity on the particle surface can be thought of as being an acid or a base, and it can have an acid dissociation or protonation constant, and a K acid and a KB, right? So losing a hydrogen ion or gaining a hydrogen ion. And these are pH dependent, obviously, just like any other acid-base reaction. So this is an example of a particle with hydroxyl groups in it. I've shown you this diagram before early on in the semester. This OH can act like an acid and become negatively charged by losing a hydrogen ion. And this might happen more so at high pH than low pH. It can also absorb a hydrogen ion from solution and become positively charged. That might happen more at low pH than high pH. In this example happens whether that OH is connected to a silicon or an aluminum or in fact an iron or a carbon. Those are the kind of four main things that can happen. And so this happens just every once in a while, that doesn't really contribute to this solvation or solubility properties for a colloid. But if we have a lot of these chemical functional groups on our particle surface, and this happens a lot, then that allows things to really charge up and go into solution. So this is an example, for instance, of taking a manganese oxide particle surface, interacting it with water in a hydration reaction, and then depending on the pH of the solution, this can either gain hydrogen ions or lose hydrogen ions net on its surface and become either a negatively charged particle or a positively charged particle. So this is a really common process. It doesn't tend to give super high density of charges, but it's the sort of fundamental process that causes charge. And now if you think back to this scenario of river water generally being acidic and seawater generally being basic, some particles will flip between this characteristic and this characteristic as a function of that change. Other particles won't. Some particles pretty much always do this regardless of the pH, or always do this regardless of the pH, but it's the ones that are kind of in between that are really interesting to us. And one of those things that's in between that's really interesting to us are iron oxyhydroxides. So this is just like a little more of the kind of chemistry of that phenomenon. You can see at low pH, we've got our metal with an OH on it, and it can attract a hydrogen ion and take a positive charge. That's kind of what this is indicating. And at high pH, it can give up this hydrogen ion and take a negative charge, right? And that has, attendant effects on the pH of the water. So that brings up this idea of something called a 0 point of charge, which has an abbreviation of ZPC. You don't have to remember that abbreviation, but just as it says, the 0 point of charge is the specific pH where the sum of the positive and negative charges on a surface are equal so that there's no net charge. Doesn't mean there's no charge, it just means that when you add up everything together, it's equal. And that's this condition here, right? You've got some amount of cations and anions attracted to that particle, and they're equal. And if we drop to a condition where the pH is lower than that 0 point of charge, also called an isoelectric point, depending on which textbook you're looking at, then we are going to tend to take a positive charge on our particle and therefore attract negative charges to it. And if our pH tends to go higher than that isoelectronic point, the opposite is going to happen. They're going to lose their hydrogen ions to take a negative charge and then therefore attract cations to them. So we can have either way, and different chemicals have different values for this. So this is just a table showing you kind of two different things. One is the actual pH of the isoelectric point or the 0 point of charge. And the other one is if we take our particle and we stick it in acid or basic medium, what kind of charge does it take? And I've kind of highlighted them with colors to show you that, for instance, these kinds of materials, which are silicate chemicals, quartz and silicon and albite and montmorillonite, kaolinite, which are clays, which we'll be talking more about, these tend to be pretty much always negatively charged. Okay, they've always lost their hydrogen ions. You can see over here some of those same minerals. You can see how low the pH has to get for them not to be negative. You got to get all the way down to a pH of 2 or less to make montmorillonite be, which is a clay, be a positive charge. That's a really very low pH. We also have these chemicals over here. These are oxides of transition metals or aluminum. So we've got iron oxide. All of these are iron oxides or oxyhydroxides. Titanium oxide. This is aluminum hydroxide and aluminum oxide, magnesium oxide. These things here, especially down here at the bottom part of the chart, pretty much always positively charged. Okay. And again, you can see those same kind of chemicals over here. But the ones that are in between, which maybe I should have used another color, I've highlighted them with red in this field, they can flip. They can change as a function of whether we're in acid or base. Should I put something to, maybe that's why this chair is always sitting there. So these are important, right? That their pH, where they flip between things, is somewhere around 6 or 7, which is exactly that pH point between river water and seawater mixing with each other. So again, as you can imagine, you have a particle surface that's got some stuff on it, it's coming down a river, and then it gets introduced into a solution where we change the particles. We can have stuff that had been stuck to the particle coming off because the particle doesn't have that charge anymore. It's flipped its charge. So these, especially iron oxide particles, the ones that have these things in red, as well as some forms of aluminum hydroxide, are also really involved in chemical changes that happen in estuaries because they can flip their sign from this acid-base reaction. Now, the other kind of charge that things can get is what we call ion exchange. So ion exchange in a simple process, it's not stoichiometric, although we can usually describe it with an equilibrium constant. It's a competition between a population of ions in solution and a particle that they might want to get stuck with, right? And so we have a particle X. And X can have some ion A stuck to it, it can have some ion B in a real world solution, it can have the whole periodic chart work of chemicals stuck to it. And they compete with each other, right? And you can write out equilibrium. And so the kind of transfer between A and B being stuck to the particle and not is an equilibrium reaction. It tends to not follow exactly the theory you might predict because of kind of non-ideal factors with the size and shape of particles and ions and so forth. It's a level of detail that we're not going to get into here because it's highly dependent on lots of different conditions. But in nature, we will find certain particles having things more commonly stuck to it and more things not commonly stuck to it. We can even take particles and move them from one solution to another and rearrange what is and what isn't stuck. That's what ion exchange is in a nutshell. So clay particles are one of the things that do this a lot. And part of the reason they do this a lot is because they're made up by these really simple structural units of polymerized silica tetrahedra, SiO4 units that are stuck together, interlayered with sheets of polymerized aluminum hydroxide, which is called gibbsite, or magnesium hydroxide, which is called brucite. These are much less common. We're not going to really talk about them very much. But these molecules are relatively open in their structure because they're flat planar things. And so it's easy for other ions to substitute in place of aluminum or silicon. And if an ion that substitutes has a different charge, it causes a charge imbalance, which then allows the particle to take on a much greater density of charge than it would from acid base. And that density of charge allows it to then attract other ions to its surface. We call them interlayer ions that will experience this reaction. Not all particles do that, but especially clay particles are really good at that. So this is an example of a clay particle. It's montmorillonite. This is how it would be written out in its ideal form, but because of substitutions when it was formed, it actually ends up being this very complicated molecule, right? Where in a spot where aluminum would be, we've substituted some iron and some magnesium. Iron +3 for aluminum +3, there's no charge imbalance there, but a magnesium +2 in the place where there would be a +3 gives us a charge imbalance. We're missing a cation, which allows another cation to stick onto its surface. And up here in the silicon layer, we find some aluminum being substituted in, which again gives the charge imbalance. And so we can see here that the exchangeable ions, which are written out here stoichiometrically, represent some fairly significant fraction of the total composition of that particle. And those exchangeable ions can change depending on the solution. So this is an example of some clays that came out of river water and some clays that came out of a dry solution. And looking at the mole fraction of sodium, magnesium, calcium, and potassium on them by exchanging them off. And we usually do that through a process of adding some other ion in massive quantities so that it outcompetes, like, barium, for instance. And then sticking them in seawater and seeing how they change from one place to the other. And you can see that, for instance, when you stick stuff into seawater, the amount of sodium that is stuck to the particles goes up, the amount of calcium goes down. And that's a fairly common characteristic of clay particles because of the distinction between salt water and average river water. And we're not so interested in these ions, these major ions, but there's a whole bunch of other chemical entities in the periodic chart that also experience this process. Some of them, when particles are changed from a riverine condition to a salty condition like seawater, will take things onto them, and other chemicals will come off of these particles. The process of things coming off we call salting off, and we see it in coastal zones and estuaries around the world, where the chemical legacy of human activities, especially radionuclides, think things like strontium-90, that are stuck to particles under one condition and then aren't stuck in another condition. Strontium being very similar to calcium, sitting just 2 elements below it in the periodic chart. So the density of charge varies with the clay composition, and it's measured by something called a cation exchange capacity, CEC. The CEC is measured at pH 7. It can change, obviously, as the pH changes, the CEC can change, but we just look at it at pH 7 to compare particles, and it's measured in milliequivalents per 100 grams. So an equivalent is a mole of charge, right? A mole of sodium ions would give you 1 equivalent. A mole of calcium ions would give you 2 equivalents for +2. So these are milliequivalents, thousandths of an equivalent, and it's in 100 grams of solid. And I think they just picked that kind of unit because it gives you sort of realistic numbers to deal with. So kaolinite, which is a kind of clay that we'll talk about weathering later, it's formed in environments where there's extensive weathering, so it doesn't get very much of this substitution on it. It's got relatively low charges on it. Come all the way down here to the bottom, vermiculite, the stuff that you put in your potting mix, has something like 30 to 10 to 30 times as much charge on it. It's got 100 to 150. This is a pretty high value, but many of these other clays are somewhere in between. That's a pretty large amount of charge, and that charge can attract ions and do all these chemical reactions that we've talked about. So the third kind of reaction is the Lewis acid, Lewis base, and this one's the most complicated, and it can work in both ways. We can have the Lewis base be our surface, and Lewis acids from solution get attracted to them, or we can have the Lewis acid be our surface and Lewis bases from solution be attracted to them. And so a couple of examples of things where this affects: we can absorb organic matter, especially negatively charged dissolved organic carbon, onto particle surfaces. We can also sorb orthophosphorus onto particle surfaces. There's lots of other things that have these kinds of interactions, but these are two of the things that are kind of important. And this is described in a fair amount of detail in the text. So let's think a little bit about this reaction in more detail. Not only does it work just like Lewis acid, Lewis base reactions in other scenarios, it's kind of like a chelation. It's just that one of the two entities, the chelating agent, the ligand, or the central ion that's being chelated is attached to a particle. So the amount that this happens depends a lot on the size of the particle. So when we look in a uniform composition of particles and we look at different size fractions, we can see how much stuff is sorbed to the particle. And we see that it goes up dramatically as the size of the particle goes down. And here we're looking at particles in wastewater. These are organic rich particles, and we're looking at the relative amount of lead, zinc, and copper. We'll talk about an example of this next time of how this works in actual waters, but most of this stuff is stuck to the smallest particle. So these are pretty complicated diagrams. I'm just going to walk you through them quickly here, but you might want to think about them and ask me questions if you have them. These are Jerome plots, right? They show you the percent from 0 to 100 of chemical entities with an acid-base relationship as a function of pH. They all go from 3 to 11. In this upper box, we've got 4 different organic solutes, right? Different kinds of carboxylic acids, along with aluminum hydroxide. Now, aluminum hydroxide can change what its conditions are, charge that it takes as a function of pH. And these solutes can take a charge difference as a function of pH, right? If the pH is very low or high, they might protonate or deprotonate. When we stick those together with the aluminum changes, I did it on its own that time, we get associations, right? And we can see here that in all these cases, we have more association mixture of these two things together when the pH is low than when the pH is high. Again, something that can happen dynamically in natural environments. In this case here, we've got iron oxyhydroxide, another one of these things that changes its charge as a function of acid or basic media. And then we've got some common anions, fluoride, phosphate, and sulfate. And what we see is that the association of the two, the sorbed solute onto that surface, for the case of fluoride and sulfate, is higher when the pH is really low. But for the case of phosphorus, there's kind of an interesting thing. Phosphorus sticks pretty much throughout. What it's sticking to is different, but this is the sum of the phosphorus. You have to get up to really high pHs, like above 9, to not have a lot of the phosphorus associated with the particle. So a lot of phosphorus that is coming off the riverine system, for instance, that we might expect to get into the ocean and feed photosynthesis in the coastal zone, doesn't, because it's stuck to iron particles, and those iron particles flocculate out in estuaries and end up in the sediments. These are just blow up those two plots. And that's what I have. God, I thought I had more content. I apologize. I guess I must have gone fast. Are there questions about this? So this, like I say, well, next time we're going to talk about, we'll give practical examples of how does this work in estuaries and why. I'm not sure what that pink thing is.
I have one question on the chart. So you said it's the acid-base equilibrium for 0 to 100. What does that mean? Does it become more acidic or more basic?
It's like, it's more like a percent. It's like if the, oh,
like
sorry.
a ratio?
Yeah, exactly. If the solution is more basic or more acidic, and you look at the percentage of all the ions, like if we just go back to this thing, right? It's saying, that will come on. Okay, so pH is across the bottom here. I just sort of accidentally cut it off here. But if you go down to a low pH, right, and you look at aluminum hydroxide, 100% of it is as this +2 form, OH2 plus, I guess I should say, until you get up to about a pH 4, and then we start getting less of this stuff and more of this stuff. And at pH 6, that's one of the buffering points. That's where the pH equals the pKa. We have an equal mixture of these two things. That's why the lines cross. Okay. So that percentage is just the relative fraction of that form of that chemical. So like this thing here, this carboxylic acid will be have its hydrogen charge associated with it, and then it will become a carboxylate. And that will happen more and more and more as the pH goes up. But at pH 4, we flip over to having more of the carboxylate and less of the carboxylic acid. And as you keep increasing the pH, once you get to a pH of 5 or so, it's 100%
Okay.
in the form of the carboxylate. So that's just a way of looking at the relative proportions of the two chemical forms as a function. It doesn't tell you why this is changing. You can happen because you're doing it in a lab, or it could happen because you're using river water and fresh water, or river water and salt water, I should say, or almost any other process that can change the pH. Okay, well, that's what I got.